Okay, so picture this. Inside every single one of your cells, there's a bustling little city. And for that city to work, you absolutely need an efficient waste management system, right? But what happens when that cleanup crew just stops showing up? Today, that's exactly what we're diving into. We're talking about a group of over 50 genetic disorders called lysosomal storage disorders.
Here's how we're going to break it all down. First, we'll get to know the cell's own recycling center. Then we'll see what happens when that system breaks down. After that, we're going to open up a few case files to really see this in action, connect the genetic dots to see how this stuff gets passed down, and finally, we'll talk about why understanding these super rare diseases is actually crucial for all of modern medicine.
All right, first things first, let's meet the star of our show today, the lysosome. Because, to really get what goes wrong, we've got to understand what's supposed to go right.
So think of the lysosome as the cell's ultimate recycling hub. Seriously, it's just packed with these super powerful enzymes. They're like tiny biological machines that just chop up and break down everything from old, worn-out cell parts to whatever garbage comes in from the outside. And this isn't just about tidying up. It's about recycling those raw materials so the cell can build new things. It is absolutely essential for keeping our cells healthy and running the way they should.
It seems like such a simple question, doesn't it? But the answer is the key to this entire group of diseases. Just one missing enzyme, one faulty worker out of hundreds, can bring the entire operation to a grinding halt.
And that brings us right to the core of the problem. When recycling fails, the trash starts to pile up. And for a cell, this isn't just messy, it's toxic. It can be completely catastrophic.
It's this three-step domino effect that defines every single one of these disorders. It always starts the same way. First, a genetic mutation means a specific enzyme, our worker, is either missing or just broken. Second, because that worker is gone, the specific material it was supposed to break down, the trash, or what scientists call the substrate, starts to accumulate. And finally, this buildup just gets toxic. It clogs the cell, messes with its function, and that's what ultimately causes the disease. Now, what's wild is that this one basic mechanism can lead to dozens of totally different diseases. It all just depends on the specific details. So let's open up a few case files and see how this all plays out in the real world. Just look at the pattern here. Each disorder is defined by a unique missing enzyme, which leads to a specific type of trash building up. So a problem with one enzyme causes Tay-Sachs, but a problem with a completely different enzyme causes Niemann-Pick. The lesson is simple, but it's so important. Different worker, different trash pile, different disease.
Let's start with a really fascinating one, eye-cell disease. Because in this case, the workers aren't actually missing. They're just getting sent to the wrong address.
So in eye-cell disease, there's a failure in the cell's postal service, basically. All these powerful lysosomal enzymes get shipped outside the cell instead of to the recycling center where they're needed. The result? The trash piles up inside the lysosome, leading to these devastating symptoms like coarse facial features and joint problems that show up in the first few months of life. It's just this perfect, tragic example of a logistics failure on a cellular level. Okay, next up, let's look at a classic diagnostic puzzle. How is it possible for two completely different diseases to share the exact same, very specific physical sign?
This is where things get tricky for doctors. Both Tay-Sachs and Niemann-Pick can cause this really distinctive cherry red spot on the back of the eye. So if you see that, how do you tell them apart? Well, you have to look for other clues. With Tay-Sachs, you don't see an enlarged liver and spleen. But with Niemann-Pick, that enlargement, that hepatosplenomegaly, is a key feature. It's exactly this kind of clinical detective work that's so essential for getting the right diagnosis.
And hey, for anyone trying to keep all this straight, here's a great little mnemonic from the source material. The A in Tay and the A in Sachs can help you remember that the deficient enzyme is hexosaminidase A, a little trick to keep in your back pocket. Now, let's take a quick look at another one, Gaucher disease. A staggering 99% of cases are the type 1 form. And what makes this one so different is that instead of showing up in babies, it often appears in adulthood, causing a really painfully enlarged spleen and serious bone problems. So, we've seen what goes wrong and where it goes wrong, but how does it happen in the first place? Well, that question takes us straight to the genetic code. The vast majority of these disorders are what we call autosomal recessive. All that means is that for a child to actually have the disease, they need to inherit one faulty copy of the gene from their mom and one faulty copy from their dad. If you only get one, you're a carrier, but you typically don't have any symptoms.
But like with any rule, there is an exception.
And that big exception is Fabry disease. Its gene is on the X chromosome, so it's inherited differently, and it primarily affects boys and men. Its symptoms are also pretty unique, starting with things like nerve pain and skin lesions, and then progressing to really serious kidney and heart problems later in life. It definitely stands out from the crowd.
Okay, I know we have covered a ton of complex biology here, so let's just zoom out for a second and ask the most important question of all. Why does studying these incredibly rare conditions matter so much?
Because at the end of the day, all of these different diseases, with all their unique symptoms and names, they all boil down to this one single shared problem. It's a breakdown in that essential cleanup crew. And studying that one glitch, well, that's incredibly powerful.
You know, when we study what happens when one tiny part of a system breaks, we learn a tremendous amount about how the whole system is supposed to work perfectly. These rare diseases are like a set of natural experiments that give us an invaluable window, a roadmap, into the fundamental workings of our own cells. And that really leaves us with a final thought to chew on. The better we get at pinpointing these tiny cellular errors, from a simple delivery mistake in eye cell disease to a single missing enzyme in Tay-Sachs, the closer we get to designing incredibly targeted, effective therapies. Understanding the problem in all its detail is always the first and most critical step toward finding a solution.